Today we will discuss about the second step of the photosynthesis that is known as the dark reaction. First of all, we will see what is dark reaction. It is the second process of photosynthesis and a series of reactions that takes place without light where carbon dioxide is fixed for synthesis of sugars. As we have seen the first process of photosynthesis that is known as the light reaction. However, when light reaction is completed, the second reaction of the photosynthesis that is known as the dark reaction or the Kelvin cycle is, is started, in which the carbon dioxide is fixed for synthesis of different types of the biomolecules. It utilizes ATPs and NADPH produced during light reaction in the absence of light. These ATPs and NADPH generally produced during light reaction, however, they reserve are stored in the lumen or stroma of the thylakoids, and then when the second process of the dark reaction is started, these ATPs and NADPH2 are used. It is also this reaction is also known as the Kelvin cycle after the name of the Kelvin Benson, the name of the scientist who discovered this cycle in the photosynthesis. It takes place in the stroma of the chloroplast. This reaction does not occur on the membrane of the thylakoid, but instead this reaction takes place in the stroma or the lumen within the chloroplast. After reacting carbon dioxide plus Obisco, the synthesis of first compound is based on the 3-carbon compound that is known as the C3 pathway. However, in other words, we can say that the dark reaction is also known as the Kelvin cycle, is also known as the C3 pathway. The C3 pathway is only known because of the first carbon compound that is initially produced after the reaction of carbon dioxide and visco, that is the 3-carbon compound, 3-phosphoglyceric acid. That's why it is known as the C3 pathway. Then mechanism of the dark reaction. The Kelvin cycle is composed of the 4 steps. Number 1, the carbon fixation, that is also known as the carboxylation. The second step is the reduction, that is also known as the glycolytic reversal. The third is the regeneration of the visco. The fourth is the synthesis of the final products. In the first step, the carbon fixation or the carboxylation. Carbon fixation means the union or the fixation of the carbon dioxide with the visco. Carboxylation means the production of the first carbon compound consisted of the carboxylic group. That is known as the carboxylation. The initial carbon dioxide acceptor is a 5-carbon compound which is known as the ribulose 1,5-bisphosphate, which is ribulose bisphosphate, which is already present in the stroma. However, this ribulose bisphosphate generally not comes from outside of the cell, but it is produced within the cells from different mechanisms already present in the cell that is utilized and combine or reaction with the carbon dioxide. So the ribulose bisphosphate combines with atmospheric carbon dioxide in the presence of the enzyme Rubisco, that is known as a ribulose 1,5-bisphosphate carboxylase. This Rubisco enzyme is present a huge amount, and some people say that the Rubisco is the enzyme of the protein, which is the highest amount in the world. Rubisco is also known as the protein that is produced in the photosynthesis mechanism. This is the almost the 90% or 95% the percentage of this protein is present in the whole of the world. However, this enzyme is responsible for combining ribulose bisphosphate with the carbon dioxide. After combining the ribulose bisphosphate and carbon dioxide, the intermediate 6-carbon compound immediately breaks down into two molecules of the 3-carbon compound, which is known as the 3-phosphoglyceric acid. The 3-phosphoglyceric acid is produced. This is the first carbon compound produced in the dark reaction. That's why this reaction or this cycle is known as the C3 cycle. The second step is the reduction. In other words, the glycolytic reversal. The ATPs and NADPs, which are produced during the light reactions, are preserved or stored within the stroma, are used during this reaction. These ATPs and NADPH2 basically are the energy storing molecules, are responsible, are used during the light dark reactions or in the absence of the light to provide the energy of the Calvin cycle. The carboxylic group of 3-phosphoglyceric acid is phosphorylated by ATP. However, the carboxylic group of the 3-PGA, in which the phosphoric acid group is provided with the ATP and produced 1,3-bisphosphoglycerate, then it reacts with the NADPH2. After the production of the 3-phosphoglyceric acid with the reaction of the 3-ATP, then the reaction or then the compound which is produced, it is known as the 1,3-bisphosphoglycerate. A 1,3, it means that two phosphate groups are present, carbon number 1 and carbon number 3 of the phosphoglycerate molecule. It means that this carbon compound, which is known as the phosphoglyceric acid, containing the 1 phosphate group. However, in case of the 1,3-bisphosphoglycerate, means that the extra 1 at carbon number 1, a phosphate group is attached. The compound is known as the bisphosphoglycerate. Bis means the two carbon compounds present at different, two phosphate groups are present at different carbon compounds, different carbons. Then it reacts with the NADPH2. Then it is reduced into 3-phosphoglyceraldehyde molecule, controlled by the enzyme triose phosphate dehydrogenase. In this equation, we can see that the 3-phosphoglyceric acid plus ATP plus NADPH2 that produced the 3-phosphoglyceraldehyde molecule, NADP plus ADP plus inorganic phosphate. However, we can see over here in this equation, in this reaction, that hydrogen ions are also released. The 1 phosphate group is also released, ADP, then there's the ATP, triphosphate, there's ADP diphosphate. 1 ATP molecule is also released. However, plus the inorganic phosphate is also released during this process. These two molecules of the phosphate groups as well as hydrogen ions are utilized for the production of the 3-phosphoglyceric acid. Then third step is the regeneration of the ribosco. The regeneration of the ribosco is very important steps in terms of the ribosco molecule, which is being utilized during the starting point of the carbon dioxide fixation. Then it needs to resynthesize because of its concentration needs to maintain within the cell. Ribosco is the starting point of the carbon dioxide fixation. It must be regenerated for its reuse in the coming reactions. The subsequent reactions of the dark reactions are the Calvin cycles. are based on the presence of the ribisco enzymes, which is responsible for the fixation of the carbon dioxide with the ribulose bisphosphate. It is regenerated in the following reactions. Uh, During the different reactions, we can see the next slide, the reactions of this phosphoglyceric acid production. The phosphoglyceraldehyde 3-phosphate are the phosphoglyceraldehyde. The 6-phosphoglyceraldehyde, or in other words, the phosphoglyceraldehyde is also known as the glyceraldehyde 3-phosphate. We can, these are the same molecules. The glyceraldehyde 3-phosphate molecules that's converted into the 6 molecules of the 3 carbon compounds that is known as the phosphoglyceraldehyde. The second, third, fourth, fifth, and sixth. These 6 phosphoglyceraldehyde molecules produced by the glyceraldehyde 3-phosphate molecule out of 6 phosphoglyceraldehyde molecules, the interconversion of the 5 phosphoglyceraldehyde molecules, the interconversion carbon number 1, 2, 3, 4, and 5. These 5 3 carbon phosphoglyceraldehyde molecules resulted regeneration of starting molecule of the ribisco. In the subsequent slide, we can see over here in the regeneration, the step of the production of the starting molecules that is known as the ribulose bisphosphate carboxylase. Over here, the 6 molecules are produced in the regeneration steps of the 3 phosphoglyceraldehyde molecules. Amongst them, the 5 molecules of the glyceraldehyde 3-phosphate molecules, the interconversion. The interconversion means that uh, these molecules convert or reaction with each other. This molecule react with each other, this molecule react with each other with the previous molecules of the PGL. This is known as the interconversion of the molecules. They produced the final product that is known as the ribulose 1,5 bisphosphate. Ribulose 1,5 bisphosphate. This is the combination. This is the final product of the interconversion of the 5 PGAL molecules. The fourth step, the synthesis of the final products. In the fourth step, out of 6 PGL molecules, only 1 PGL molecule comes out of the cycle. The 1 PGL molecule comes out of the cycle. That is responsible for the synthesis of different types of the biomolecules. That is the glucose, starch, and other building blocks of the cells. However, in details, the following compounds are synthesized by one molecule of the PGAL. The number 1 is the carbohydrates. In carbohydrates, the triose sugar combines to form the glucose and fructose. The molecules of glucose combines to form the starch and the cellulose. Starch and the cellulose. Cellulose is present in the plants as well as starch is present in the plants, whereas the glycogen is present in the animals. However, the composition of all the cellulose as well as the starch molecules is the combinations of many glucose molecules, only one type of the molecules. Second compound which is produced during the dark reaction is the lipids. Some triose phosphate molecules are also converted into acetyl coenzyme A, then converted into the fatty acids. The acetyl coenzyme A is responsible for synthesis for starting of the Krebs cycle during the respiration process. However, this molecule acetyl coenzyme A is responsible for the synthesis of the fatty acids by reaction with the triose phosphates. The third one is the proteins. The triose phosphate molecules is also responsible for the conversion of the proteins by using the alpha ketoglutarate in the Krebs cycle. During the respiration process, the alpha ketoglutarate combines with the ammonium ions to form the amino acids. Of these amino acids are the building blocks of the proteins. The other compound that is known as the DNA, RNA, and some other biomolecules are also synthesized during this process of the dark reactions. It means that the dark reaction is the reaction in which the first three carbon compound is produced, that is known as the 3-phosphoglycerate. That's why it is known as the C3 pathway. In this process, the six molecules of phosphoglyceraldehyde are produced. Amongst them, the one molecule of phosphoglyceraldehyde is utilized for the synthesis of different types of the biomolecules, which is carbohydrates, lipids, proteins, DNA, RNA, etc., and other biomolecules. However, the five phosphoglyceraldehyde molecules with interaction, the reaction with each other, they produces different types of the molecules and subsequently produce the final molecule that is known as the 3-phosphoglycerate. The ribulose bisphosphate, sorry, ribulose bisphosphate. Ribulose bisphosphate is the molecule once produced is stored within the stroma, and one carbon dioxide comes combined with the ribulose bisphosphate in the presence of the ribisco and start a new Calvin cycle or the C3 pathway.